For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Problem 1. Sugar isomerization A. Convert alpha-glucose to its furanose form. B. Is it more likely for it to be found in the cyclized or linearized form? Answer. Cyclized. C. Draw the linear depiction of D-glucose. Answer. See picture above. Problem 2. Synthesis. I would like to make D-galactose, shown, from D-fructose, also shown. Show which steps and enzymes are necessary for this. Enzymes you can use: ketosaldose, isomerase, epimerase. Problem 3. Sugar characteristics and nomenclature, from previous exam. A. Answer the following by circling the numbers corresponding to the sugars above which best answer each question. There may be 0, 1, or 1 answer for each question. Provide brief explanation only for the last question where requested. Which sugars is /are a pyranose? Which sugars is /are drawn as an alpha-pyranose or as an alpha-furanose? 3 of V. Identify all the aldose sugars. 2, 3, V. Identify all the reducing sugars as. 2, 3, 4, V. Which sugars is /are not found in nature? Briefly explain your answer. 2. Nature uses D sugars, not L sugars. B. Name the following sugar. Alpha-deribofuranocil 2 to 4, beta-dixylofuranose. C. Draw alpha-diglucopyranocil 1 to 3, diglyceraldehyde. D. Specify which of the sugars named /drawn in parts B and C above are reducing sugars. If neither is a reducing sugar, make this clear. Briefly explain your answer. Answer. Both sugars are reducing since both have an accessible aldehyde or ketone. Problem 4. Chemistry of glycolysis. A. The reactants in the net reaction of glycolysis are given below. Complete the reaction by writing the products and balancing the equation, including charges. Assume that all inorganic phosphate pi is in the form HPO42-. Glucose C6H12O6 plus 2 ADP plus 2 pi plus 2 NAD plus 2 pyruvate C3H3O3 plus 2 ATP plus 2 NADH plus 2 H2O plus 2 H plus. B. One component of the net reaction of glycolysis is the synthesis of ATP from ADP and pi. For each of the following reactions, write the balanced chemical equation. Assume that all inorganic phosphate pi is in the form HPO42-. Which step of glycolysis consumes pi? Which step uses that same pi to generate ATP? For each step, name the enzyme and draw the balanced reaction using names, formulas, or structures of the reactants and products. 1. The synthesis of ATP from ADP and pi, aka reverse of ATP hydrolysis. Answer. ADP plus pi plus H plus ATP plus H2O. 2. The step of glycolysis that consumes pi. Answer. Ga3P plus pi plus NAD plus 1,3-BPG plus NADH plus H plus. 3. The step of glycolysis that makes ATP from a product in question 4b2. Answer. 1,3-BPG plus ADP3PG plus ATP. C. Red blood cells express a special enzyme, 2,3-bisphosphoglycerate mutase, that turns 1,3-bisphosphoglycerate, 1,3-BPG, into 2,3-bisphosphoglycerate, 2,3-BPG. You may recall that 2,3-BPG stabilizes the T state of hemoglobin and decreases its oxygen affinity. 2,3-BPG re-enters glycolysis when it is dephosphorylated by 2,3-BPG phosphatase to 3-phosphoglycerate, 3-PG. These two reactions are named the Lubering-Rappaport pathway. 1. If a glucose molecule enters glycolysis, and both of the 1,3-BPG molecules it yields go through the Lubering-Rappaport pathway instead of through phosphoglycerate kinase, what is the net number of ATP produced by glycolysis of this molecule of glucose? Answer: 0. 2. Genetic defects in glycolysis can disrupt levels of 2,3-PBG and diminish the ability of red blood cells to transport oxygen. How would 2,3-BPG level and hemoglobin oxygen affinity be affected by defects in the following enzymes? Enzyme 2,3-BPG +//0, HbO2 affinity +//0. Hexokinase plus. Pyruvate kinase plus. 3. In 2006, scientists discovered that cells in the outermost layer of the placenta also express 2,3-bisphosphoglycerate mutase. 2,3-BPG binds more tightly to adult hemoglobin than to fetal hemoglobin. Why would expressing this enzyme in the placenta help the fetus get oxygen? Answer: production of 2,3-BPG in the placenta would cause the mother's blood to release oxygen to the fetus in the vicinity of the placenta, and the fetus's blood would absorb the oxygen because 2,3-BPG does not reduce the oxygen affinity of fetal hemoglobin as much as adult hemoglobin. D. Organisms growing anaerobically cannot perform glycolysis for long without reducing the pyruvate from glycolysis into another compound, most commonly to lactate or to ethanol plus CO2. Both of these reactions are given below in their unbalanced forms. Explain in one sentence why one of these reducing steps is needed to sustain anaerobic glycolysis. 1. Pyruvate plus NADH lactate plus NAD plus 2, pyruvate plus NADH ethanol plus CO2 plus NAD plus. Answer: this step is required to replenish the NAD plus that glycolysis consumes. In mammals, muscles produce lactate during intense exercise because glucose availability for glycolysis is in excess of oxygen delivery to support complete glucose oxidation. Lactate is transported via the bloodstream to the liver, where it is converted back to pyruvate and then to glucose via gluconeogenesis, which returns to the muscles, the Cori cycle. Write the balanced reaction that converts lactate back into pyruvate in the liver. Answer: lactate, C3H5O3, plus NAD plus pyruvate, C3H3O3, plus NADH plus H plus. Problem 5. Energetics of glycolysis. A. The complete combustion of glucose, C6H12O6, in oxygen, O2, yields water and CO2. Write the balanced reaction for the combustion of glucose. Answer: glucose, C6H12O6, plus 6O2 6H2O plus 6CO2. B. The free energy, increment g degree, of this reaction under standard conditions, 25 degrees Celsius, each species at 1 m, is minus 2930.4 kJ/mol, equilibrator.Weitzman.ACIL. Calculate the free energy under physiological conditions, assuming that T equals 37 degrees Celsius, glucose equals 1 mm, O2 equals 2.0 mm, CO2 equals 2.5 mm. C. Assume that under physiological conditions, hydrolysis of ATP has an increment G of minus 46 kJ/mol. What percentage of the chemical energy in glucose, your answer to 5b, gets stored in the molecules of ATP produced from glycolysis? Does glycolysis by itself seem like an efficient way to get energy from glucose? 2 * -46 / -2909 equals 3.2%.